Hi everyone, my name is Mia Martinez and I'm here to talk to you about the research I've done this past semester in Dr. Chopra's lab. Dr. Chopra runs a neuroimmunology lab, which is combining neuroscience and immunology, so specifically looking at the immune system of the brain. Dr. Chopra's lab focuses on Alzheimer's, so taking a look at what causes Alzheimer's. One of the primary cells in the brain is called microglial cells, and Dr. Chopra's lab has the hypothesis that Alzheimer's is caused by the accumulation of lipid droplets on these microglial cells, effectively methylating the microglial cells. So they accumulate around these cells and then repress their function, leading to Alzheimer's. Dr. Chopra's lab has a few different sections, and I have gotten the privilege to work in both sort of biology side as well as the chemistry side. So on the biology side, Western blots. I've had the privilege to be there as new Western blot instrumentation was being implemented and kind of being right in the middle of learning how to do this new process. So in this top left picture here, you'll see we have sort of bunch of purple little squares here in this gel. So that is pipetted samples that have been mixed with a dye in order so that we can see them in the gel. That so this gel is inserted into a gasket, and in this it's sort of in a bucket filled with a buffer. So this will allow these gels to run down through, as you'll see in this middle picture here, the gels run down, the protein samples will run down through the gel after going through a process of electrophoresis, which is when we set these up and run electricity of 200 volts through this gel for 20 minutes. So this buffer carries these samples almost sort of creating a chromatography. But so it shows what proteins are in each. So the lines we have in this bottom picture are the samples. So that is showing you, that's a sample line. So a kind of comparison chart to run. And so once we run through these and the electrophoresis is complete, what we then have to do is transfer this gel onto a membrane because the gel is very delicate, whereas the membrane is a bit more stable. So layer the gel between a sponge into a membrane and then place it in this cassette. So as you can see, we have our gel laying on a sponge in an open cassette, and that cassette is then closed and then placed into a compression machine to allow the transfer. This is part of what I've done on the biology side. Now going to the chemistry side, we focus sort of on the EDC reaction. So part of the research that Dr. Chopra is doing is tracking the activity of the microglial cells. Doing this by attaching a dye to the microglial cells in order to track the activity, be able to see the microglial cells within the brain. So the dye we're using is rhodamine B. However, the issue is that the microglial cells don't have an attachment point for the rhodamine B. There's no way for them to attach. So what we do is a sort of coupling reaction, an EDC reaction. So EDC is 1-ethyl 3,3-dimethylpropyl carbonic. And so taking this and using it as a sort of not catalyst so much, but a prompter for this reaction, an intermediate, so to speak. We take our rhodamine B dye and react it with a boronic acid, and then inside an EDC reaction. So the EDC that we chose to use was DCM. So as you can see on the left here, it's lots of different symbols, but essentially we have our rhodamine B and our boronic acid reacting with our DCM to then produce a rhodamine B sample that can be paired with the boronic acid. So over here on the right, we have our rhodamine B and our boronic acid reacting to form this now attachable form that can allow us to track the microglia. So here in the middle is the purification of the DCM. So DCM made in-house, we then have to purify it. So we're using a water workup extraction to remove these impurities from DCM. And because we have such a large amount, we're using a titrate flask and water. So water and DCM are immiscible and they have different densities. So water is going to sit on the bottom, which is why we're using a titrate flask to then drop down into a beaker containing sodium sulfate. So as we run through and we open up the spout, there we go. We open up the spout to allow the water to run through that has mixed with the DCM and kind of attached to the impurities. And so the water will drop through before the DCM ever reaches the tip of our flask here, and the water will drop down into the sodium sulfate, which will then be absorbed. So we're left with a very pure form of DCM. And so we keep adding this. If we were doing this with a smaller thing, we would a smaller amount of DCM, we would want to use ethyl acetate and it kind of reversed in a test tube so that the ethyl acetate was on top, so we could use a pipette and pull that out. So then using that DCM that we've purified, we now can do the coupling, which is going to be done in the next few weeks, to pair the rhodamine B with the boronic acid, and then moving forward, we can attach that to the microglial cells to be able to track. So I've had the great opportunity to work with these fabulous people in the lab and be exposed to all these things. Thank you.